Hello there, my friend. I'm really glad you're here tonight. The world can be a bit loud sometimes, can't it? So before we start, let's just pause for a moment. Take a slow breath in and let it out. If this is your first time here, you can always come back whenever the day feels too heavy. This little place will be waiting for you, quiet, familiar, and kind. And if you've been here before, thank you for returning. It's a strange kind of comfort, isn't it? To know that somewhere out there, someone else is also listening to the same story under the same night sky. Tell me, when was the last time you looked at the sea and wondered what lives beneath it? Not the waves, not the fish, but the world that's quietly building itself, piece by piece in silence. I'd love to know what the ocean makes you think of. Now, let's drift together into that world. A place of soft light and slow time, where mountains are built by creatures no larger than your fingertip, and where the sea keeps its oldest secrets hidden in plain sight. Tonight's story is about coral, the living architects of the ocean. The coral awakens. Coral reefs are among the most remarkable and complex ecosystems on Earth. Although they cover less than 0.1% of the ocean floor, they provide a home, directly or indirectly, for nearly a quarter of all known marine species. To understand how such vast ecosystems exist, we must begin with the coral itself, a small and surprisingly fragile animal. Many people think of coral as a type of rock, or perhaps a kind of underwater plant. This misunderstanding is easy to make. Corals remain fixed in one place. They appear rigid, and they often resemble branches or stones. But in fact, corals are animals, living organisms that belong to the phylum Cnidaria, the same group as jellyfish and sea anemones. Like their relatives, corals possess specialized stinging cells used for defense and feeding. Each coral organism is called a polyp. A single coral polyp is only a few millimeters to a few centimeters in diameter. Its body is soft, cylindrical, and simple in structure. At the center is a mouth surrounded by tentacles. These tentacles are covered with nematocysts, microscopic capsules containing coiled, barbed threads that can release toxins. When small plankton or organic particles drift by, the polyp's tentacles fire these nematocysts to capture them. Once caught, the prey is brought to the mouth, digested in a central cavity, and the nutrients are shared across the entire colony. A coral colony is not a single animal, but a community of thousands of polyps connected by thin layers of tissue. This connection allows for the exchange of nutrients and signals between individuals. If one polyp captures food, neighboring polyps can absorb part of the nutrients. The colony behaves almost like a single, multicellular organism, each unit working for the stability of the whole. The foundation of a coral's life is its skeleton. Through biochemical processes, each polyp secretes calcium carbonate, a mineral that hardens around its base. As new polyps form, they continue to build upon the skeletons of previous generations. Over time, this slow and steady accumulation produces the massive limestone structures that we recognize as coral reefs. The mineral form used by corals is primarily aragonite, a crystalline form of calcium carbonate that is denser and more soluble than the calcite found in most seashells. The process of skeleton formation, known as calcification, depends on several environmental factors: temperature, light, salinity, and the concentration of carbon dioxide in seawater. Corals grow fastest in clear, warm, shallow water with stable conditions. Most reef-building species thrive in temperatures between 23 and 29 degrees Celsius, and in salinity levels close to that of normal seawater, around 35 parts per thousand. Even small changes outside this range can stress or kill the coral. There are two main types of coral: hard corals and soft corals. Hard corals, also called stony corals or scleractinia, are the primary builders of coral reefs. Their rigid skeletons give reefs their solid structure. Soft corals, on the other hand, such as sea fans, sea pens, and sea whips, lack a heavy mineral skeleton. They contain flexible protein fibers called gorgonin, which allow them to move with the current like underwater plants. Although soft corals do not build reefs, they still play an important role in the ecosystem by providing habitat and filtering nutrients from the water. Hard and soft corals often coexist in the same area, forming complex environments that host thousands of species. Within a few square meters of a healthy reef, scientists may find dozens of coral species and hundreds of species of fish, crustaceans, worms, and mollusks. The variety of forms, branching, massive, encrusting, or plate-like, creates endless hiding places and feeding zones. Every shape has a function, and together they form one of the most biodiverse systems on the planet. A key to the success of corals is their relationship with microscopic algae called zooxanthellae. These single-celled algae live inside the coral's tissues, forming a symbiotic partnership. During daylight, the algae perform photosynthesis, using sunlight and carbon dioxide to produce sugars and oxygen. The coral absorbs these sugars as its primary source of energy, allowing it to thrive even in clear, nutrient-poor tropical waters. In return, the algae receive protection within the coral's cells and benefit from the coral's waste products, such as carbon dioxide and nitrogen compounds. This mutualistic relationship is delicate but efficient. It allows corals to occupy regions where food is scarce, turning sunlight into the foundation of an entire food web. The presence of zooxanthellae also gives corals their color. Different pigments within the algae produce hues of brown, yellow, green, and red. Without the algae, the coral's transparent tissue reveals its white skeleton beneath. This is what scientists call coral bleaching. When environmental stress such as high temperature or pollution occurs, corals may expel their algae. If the stress lasts only a short time, the coral can recover by taking in new algae. If not, the coral starves and eventually dies. This process, although visually striking, is a clear sign of ecological imbalance. Corals are highly dependent on light, so most reef-building species grow at depths shallower than 30 meters. In deeper or darker areas, where sunlight is weaker, different types of corals live without algae and rely entirely on capturing food from the water. These deep water corals can survive in cold and dark conditions, sometimes at depths greater than 2,000 meters, and can be found in places such as Norway, the North Atlantic, and even Antarctica. Unlike tropical corals, they grow very slowly and do not form colorful reefs. Growth rates among corals vary greatly. Fast-growing branching corals, such as Acropora, may extend several centimeters per year. Massive dome-shaped corals, like porites, may grow only a few millimeters annually. Despite their slow pace, over centuries and millennia, these corals can form structures that reach several meters in height and stretch across vast distances. Coral reefs form primarily in tropical and subtropical waters between 30 degrees north and 30 degrees south of the equator. Here, the combination of sunlight, stable temperature, and clear water creates ideal conditions for growth. The largest of all is the Great Barrier Reef in Australia, which extends for over 2,300 kilometers and is visible from space. Other major reef systems include the Coral Triangle in Southeast Asia, the Mesoamerican Barrier Reef in the Caribbean, and the Red Sea reefs along the coasts of Egypt and Saudi Arabia. In addition to reproduction through budding, corals also reproduce sexually through the release of eggs and sperm into the water column, a process known as broadcast spawning. Many coral species synchronize this event with lunar cycles and water temperature. Once fertilized, the eggs develop into tiny, free-swimming larvae called planulae. After drifting for several days or weeks, these larvae settle on a suitable surface and transform into new polyps, beginning the cycle again. Each living coral reef is a layered record of life and time. Beneath the living surface lies the skeletons of countless generations that came before. These layers can be studied like tree rings, allowing scientists to reconstruct past climates, sea levels, and even patterns of ocean chemistry. Coral skeletons contain isotopes of oxygen and carbon that record the temperature and composition of seawater at the time they formed. In this way, corals act as natural archives of Earth's environmental history. Although each polyp is tiny and fragile, the collective effort of millions of these organisms creates a structure strong enough to resist waves and storms. The reef protects coastlines from erosion, provides shelter for marine life, and supports fisheries and tourism that sustain hundreds of millions of people worldwide. To the human eye, a coral reef may look like a single, still formation. But scientifically, it is one of the most active biological systems known. Every surface is occupied by algae, sponges, crustaceans, mollusks, worms, and fish, all interacting in cycles of feeding, cleaning, and rebuilding. The corals themselves are at the center of this activity, quietly maintaining balance through a combination of biological precision and environmental cooperation. Understanding the coral is to understand patience. Each structure we see today represents thousands of years of uninterrupted growth and biological teamwork. The reef is not built overnight, and not by force, but by constant, microscopic effort, layer upon layer, generation after generation. In the next chapter, we will examine how corals become architects of the sea, how their growth shapes coastlines, influences ocean currents, and provides the foundation for entire marine ecosystems. Architects of the sea. Corals are often called the architects of the ocean. This title is not poetic exaggeration, but a scientific description of their role in shaping marine landscapes. Through slow biological construction, coral colonies form vast geological structures known as reefs. Massive living fortresses built from calcium carbonate. Over centuries and millennia, these reefs create the foundation for entire ecosystems, coastlines, and even islands. To understand how corals build these environments, it is important to see them not as individual organisms, but as biological engineers working on a scale far beyond their size. A coral reef begins with a single polyp attaching to a solid surface, often a piece of rock, a shell, or an old coral skeleton. Once settled, it divides and multiplies through asexual budding, forming a small colony. As the colony grows, each new generation of polyps deposits calcium carbonate beneath itself, creating a thin layer of rock. Over time, these layers accumulate, and neighboring colonies merge, forming a continuous structure. This process happens very slowly. Depending on the species and environmental conditions, a coral reef may grow between 1 and 15 centimeters per year. When sea levels rise or fall, the corals adjust their growth direction to remain within the ideal depth range for light. Through this balance of biology and geology, reefs evolve in shape and height, adapting to the environment around them. There are three main types of coral reefs identified by scientists. The first type is the fringing reef, which grows directly along the shoreline. Fringing reefs are common in regions like the Red Sea and parts of Southeast Asia. They begin as narrow platforms near the coast and gradually expand seaward as the coral continues to build. The second type is the barrier reef, which forms further offshore, separated from land by a lagoon. Barrier reefs can extend for hundreds of kilometers. The Great Barrier Reef of Australia is the largest and best-known example, stretching more than 2,300 kilometers and consisting of about 3,000 individual reef systems. The third type is the atoll. Atolls are ring-shaped reefs that encircle a central lagoon but have no land in the middle. They often form when a volcanic island slowly sinks below the surface of the ocean, while the surrounding coral continues to grow upward. This process was first explained by Charles Darwin during his voyage on the Beagle. He proposed that as the volcanic island subsides, the coral reef remains at sea level, eventually leaving behind a circular formation, a natural monument to both geology and biology. A coral reef is not uniform. It has distinct zones shaped by wave energy, light, and depth. The reef crest is the highest part, often exposed to waves and spray. Here, corals are compact and sturdy, built to withstand strong physical forces. The forereef lies on the seaward side and slopes down into deeper water. It hosts massive coral formations and diverse fish populations. Behind the crest is the reef flat, a broad and relatively shallow area, sometimes covered by seagrass or small colonies of corals adapted to high sunlight and heat. Further inside is the lagoon, where water is calmer and sediment accumulates. Each zone supports its own community of organisms, adapted to different light levels, water flow, and nutrient conditions. This spatial organization allows the reef to function like a self-regulating system. Energy from waves is absorbed by the outer crest, protecting the more delicate ecosystems inside. This natural barrier reduces coastal erosion, making coral reefs essential for the protection of shorelines and human settlements. Coral reefs are among the most densely populated ecosystems on Earth. A single square meter of reef can contain hundreds of species, each playing a specific role in maintaining ecological balance. Fish, invertebrates, and algae interact constantly in cycles of feeding, cleaning, and sheltering. Small fish find protection within coral branches, while larger predators patrol the open water. Many reef fish have evolved specialized behaviors to coexist with corals. For example, cleaner wrasses remove parasites from bigger fish, and parrotfish graze on algae that could otherwise overgrow coral surfaces. Parrotfish also contribute to the reef's sand production by grinding coral skeletons as they feed and excreting fine, white sand. A single large parrotfish can produce several hundred kilograms of sand each year. Other organisms also play critical roles. Sea urchins control algal growth by scraping rocks and dead coral. Sponges filter water, removing bacteria and recycling nutrients. Crustose coralline algae act as a natural cement, binding coral fragments together and stabilizing the reef's structure. This balance between building and erosion ensures that the reef remains dynamic rather than static. If one group becomes too dominant, the system destabilizes. For example, when sea urchin populations decline due to disease or overfishing, algae can grow unchecked, covering the coral and blocking sunlight. Similarly, outbreaks of the crown-of-thorns starfish, a natural predator of coral polyps, can devastate large areas of reef if their numbers rise too high. Healthy reefs maintain equilibrium through a constant competition among species. Reef development is controlled by several environmental factors. The most important are light, temperature, water quality, and sedimentation. Light is essential because most reef-building corals rely on photosynthesis through their symbiotic algae. Therefore, corals grow best in clear, shallow waters where sunlight penetrates easily. Temperature plays an equally important role. If the water becomes too cold or too hot for an extended period, the coral-algae partnership breaks down, resulting in bleaching. While some coral species can tolerate small fluctuations, prolonged heat stress can cause large-scale mortality. Sediment and pollution also affect coral health. Excess nutrients from agriculture or sewage encourage algal blooms, which can outcompete corals and reduce oxygen levels. Fine sediments can settle on coral surfaces, blocking sunlight and interfering with feeding. Even minor changes in water clarity can reduce photosynthetic efficiency and slow growth. Corals prefer stable, low-nutrient environments. Yet these conditions are increasingly rare near human-populated coasts. Understanding the physical limits of coral growth helps scientists predict which reefs are most at risk. The growth of a reef, known as accretion, is always balanced by processes of erosion. While corals and algae deposit calcium carbonate, other organisms break it down. Boring sponges, worms, and mollusks dig into the rock, creating tunnels and weakening structures. Waves and storms physically erode fragile coral branches. Herbivorous fish and sea urchins further grind coral debris into sand. This cycle of building and breakdown is continuous. A healthy reef maintains balance. The amount of calcium carbonate produced equals the amount lost. If environmental stress reduces coral growth, erosion can exceed accretion, and the reef begins to degrade. This delicate equilibrium determines whether a reef expands or collapses over time. Beyond their ecological importance, coral reefs play a crucial physical role in protecting coastlines. By absorbing wave energy, they reduce erosion and flooding during storms. Studies show that reefs can dissipate up to 90% of wave force before it reaches the shore. This makes them natural breakwaters that protect beaches, mangroves, and human settlements. The destruction of reefs can therefore have direct consequences for people. Without reefs, waves strike the coast with greater intensity, causing loss of land, infrastructure, and sometimes lives. In many tropical regions, coral reefs are the first and strongest line of defense against storm damage. Coral reefs also serve as nurseries for marine life. Many species of fish and invertebrates spend the early stages of their lives in the protection of coral structures before moving to open water. The complex shapes of the reef, with its caves, ledges, and crevices, provide shelter from predators. Juvenile fish can hide in small spaces where larger animals cannot reach. Because of this, the health of coral reefs directly affects nearby ecosystems such as seagrass meadows and mangrove forests. These systems are interconnected through the movement of water, nutrients, and organisms. When one is damaged, the others suffer as well. At the biochemical level, coral reefs are central to oceanic carbon cycling. Through calcification, corals remove carbon dioxide from seawater and store it in solid form as calcium carbonate. However, this process also releases some CO2 back into the atmosphere, making coral reefs both a sink and a source of carbon. The balance between these two roles depends on the overall health of the reef. In addition to carbon cycling, corals contribute to nutrient recycling. They capture nitrogen and phosphorus from organic matter and release them in forms usable by other organisms. This tight recycling of nutrients supports high productivity despite the low nutrient levels of tropical waters. In this way, coral reefs function as biological engines, constantly processing and redistributing energy and matter within the marine environment. over geological time. Coral reefs influence the shape of continents and ocean basins. When coral growth keeps pace with sea level changes, reefs can rise continuously, forming thick deposits of limestone. These deposits can later become part of the land as tectonic uplift exposes them above sea level. Much of the world's tropical limestone and karst landscapes originated from ancient reefs that formed millions of years ago. Coral skeletons found in these rocks provide valuable records of Earth's past climate. Scientists study the chemical composition of fossilized corals to reconstruct sea temperatures and ocean chemistry from different geological periods. Such data are essential for understanding long-term climate trends and predicting future changes. Every reef represents the outcome of balance between growth and erosion, light and darkness, stability and disturbance. No part of a reef exists in isolation. Each coral colony depends on the species around it, the movement of water above it, and the chemical composition of the sea itself. This interdependence is what makes reefs resilient, yet also what makes them vulnerable when one element fails. The architecture of a coral reef is therefore both biological and environmental. It is built not only by coral polyps, but also by currents, tides, and waves that shape its structure over time. A reef is a product of cooperation between living organisms and physical forces, a collaboration that can take thousands of years to complete. When viewed in this way, the term architects of the sea becomes literal. Corals design and construct their environment, not with intention, but through collective biological activity. They create habitats for others, protect coastlines, and alter geological formations. Through the steady accumulation of their skeletons, they transform the seafloor into one of the most productive systems on the planet. Symphony of light. Light is the primary source of energy for life on Earth, and the same is true beneath the surface of the ocean. In the shallow seas where coral reefs form, sunlight penetrates the water, providing the energy that drives one of the most efficient biological partnerships known in nature: the relationship between coral animals and microscopic algae. This interaction between light and life defines every aspect of a reef's existence. Sunlight determines where corals can live. Reef-building corals depend on light because of their symbiotic algae known as zooxanthellae. These algae perform photosynthesis, the process of converting sunlight, carbon dioxide, and water into sugars and oxygen. The energy produced through this process supports not only the coral itself, but also the thousands of species that rely on the reef for food and shelter. The depth to which sunlight can reach in seawater depends on the clarity of the water and the angle of the sun. In very clear tropical waters, light can penetrate to a depth of about 100 meters, but the intensity drops rapidly with depth. Most reef-building corals live within the upper 30 meters, where light intensity is strong enough for photosynthesis. Beyond this zone, light becomes too weak, and only specialized corals and organisms adapted to darkness can survive. The wavelength composition of sunlight also changes underwater. Red and orange wavelengths are absorbed quickly, while blue and green penetrate deepest. As a result, the underwater environment takes on a bluish color. Corals and algae have evolved pigments that capture light efficiently under these conditions. Different species adapt to specific light intensities and depths, creating layers of biodiversity based on how each organism uses light. At the core of the reef's energy system is the partnership between coral polyps and their symbiotic algae. Each coral polyp contains thousands of microscopic algal cells within its tissues. These algae, belonging to the genus Symbiodinium and related lineages, perform photosynthesis and supply the coral with up to 90% of its energy needs. In return, the coral provides the algae with a stable environment, a safe place protected from predators, and access to the coral's metabolic waste, which contains carbon dioxide, nitrogen, and phosphorus, essential nutrients for photosynthesis. This relationship is mutually beneficial and highly specialized. Inside the coral's cells, the algae exist in tiny vacuoles called symbiosomes. The exchange of nutrients between coral and algae is regulated by chemical signals. During the day, when sunlight is available, photosynthesis occurs at high rates, producing oxygen and carbohydrates. The coral absorbs these products and converts them into lipids, proteins, and calcium carbonate. At night, when photosynthesis stops, the coral continues to respire, using stored energy and releasing carbon dioxide back into the algae once daylight returns. This cyclical exchange forms a tightly coupled metabolic system. It allows coral reefs to thrive in nutrient-poor waters where other ecosystems would struggle to survive. Photosynthesis in seawater follows the same basic principles as on land, but the physical conditions are different. Underwater, light is scattered and absorbed in complex ways. The algae must capture limited light efficiently, and they do so using specialized pigments such as chlorophyll A, chlorophyll C2, and peridinin. Peridinin, in particular, allows the algae to absorb blue-green light, the dominant wavelength at depths where corals live. These pigments give corals their characteristic brownish or golden coloration. In some species, additional fluorescent proteins within the coral tissue modify the light spectrum, producing vivid hues of green, pink, and red. These pigments can protect the coral from excessive sunlight by acting as filters, or can enhance photosynthesis by redistributing light within the tissue. The efficiency of this light use is extraordinary. Even in low nutrient conditions, a single square meter of healthy reef can produce as much organic material as a fertile tropical forest on land. This productivity supports a vast web of life extending far beyond the corals themselves. Corals and algae form the base of the reef's energy pyramid. The sugars produced by photosynthesis are used directly by the coral for respiration and growth. Excess organic matter, in the form of mucus and dissolved compounds, is released into the surrounding water. This material feeds bacteria, plankton, and filter-feeding organisms like sponges. In this way, energy flows outward from the coral in multiple directions, linking microscopic processes to large-scale ecological dynamics. Sponges, for instance, play an important role by converting dissolved organic matter into particles that can be consumed by other animals, a process known as the sponge loop. This efficient recycling system keeps nutrients circulating within the reef, allowing it to remain productive even in waters that are otherwise poor in nutrients. The balance of this energy exchange is delicate. If environmental stress reduces photosynthetic efficiency, the coral's energy intake declines, making it more susceptible to disease, bleaching, and death. Conversely, when conditions are optimal, the reef becomes a self-sustaining ecosystem capable of supporting dense populations of life. The coral-algae relationship follows a clear daily rhythm. During daylight, photosynthesis dominates. Coral polyps retract their tentacles to avoid shading the algae, allowing maximum exposure to sunlight. As oxygen and sugars accumulate, the coral's metabolic activity increases, and calcium carbonate deposition reaches its peak in the afternoon. At night, photosynthesis stops, and the coral switches from a phototrophic to a heterotrophic mode. It extends its tentacles to capture plankton and organic matter from the water. This nightly feeding supplements the energy provided by the algae, especially in times of low light or stress. This dual strategy, combining photosynthesis and active feeding, gives corals flexibility to survive in varying environmental conditions. Not all coral reefs receive the same amount of light. Some grow in shallow lagoons exposed to intense sunlight, while others exist at the edge of the photic zone where light is dim. To adapt, both corals and their symbiotic algae can adjust to different light intensities. This process is known as photoacclimation. In bright environments, algae reduce their pigment concentration to prevent light stress, and corals produce fluorescent proteins that act as sunscreens. In low light conditions, algae increase their pigment density and rearrange chloroplasts to capture as much light as possible. These adjustments can happen within days or weeks, showing how dynamic coral-algae interactions are at the cellular level. Some corals also alter the density or type of algae living within them. Different species of Symbiodinium vary in their tolerance to light and temperature. Corals that host more heat-tolerant strains can survive in warmer, brighter environments, though sometimes at the cost of slower growth. As light fades with depth, the rules of reef life change. Beyond about 30 to 40 meters, most reef-building corals cannot grow effectively because there is not enough light for photosynthesis. However, specialized communities exist even in these low-light zones. Certain corals and algae have evolved highly efficient pigments and large surface areas to maximize light capture. Water clarity also determines the health of a reef. Sediments, plankton, or pollution particles in the water can block sunlight, reducing photosynthetic rates. In coastal regions affected by human activity, increased turbidity often leads to coral decline. This is why reefs thrive best in clear, oligotrophic waters, where the light can penetrate deeply without obstruction. Too much light can be as harmful as too little. When corals experience heat stress or excessive radiation, photosynthesis within the algae becomes inefficient. This causes the production of reactive oxygen molecules, which damage both the algae and the coral's tissues. To protect themselves, corals expel their algae, losing their main source of energy and color. This event is called coral bleaching. Bleaching does not always mean immediate death. If temperatures return to normal, corals can reabsorb algae or acquire new symbionts. However, prolonged or repeated bleaching weakens the coral, reducing reproduction and growth rates. Severe bleaching events have increased in frequency over the past decades, largely due to rising sea temperatures caused by climate change. Scientists monitor degree heating weeks, a measure of how long and how much the temperature exceeds normal levels, to predict bleaching risk. Even a few degrees above the typical summer maximum, sustained for several weeks, can trigger mass bleaching across entire regions. On a microscopic scale, coral tissues create complex light fields. The surface of a coral is not smooth. It is covered with ridges, folds, and layers that scatter light in many directions. This scattering increases the total amount of light available to the algae. Some corals also have internal light guiding structures that distribute illumination to shaded parts of the colony. Researchers use fiber optic probes and microsensors to study how light moves through coral tissues. These measurements show that corals can focus or diffuse light, depending on the geometry of their skeleton. This optical adaptation maximizes energy capture and contributes to the coral's overall efficiency. Such fine-scale adaptations explain why corals can grow successfully even under fluctuating light conditions, such as in shallow waters where sunlight is reflected and refracted by waves. The influence of light extends beyond corals themselves. Many reef organisms, including algae, sponges, and fish, respond to daily and seasonal cycles of illumination. Light regulates reproduction, migration, and feeding patterns across the entire ecosystem. For example, some corals synchronize their spawning events with lunar cycles, ensuring that eggs and sperm are released when currents and tides are most favorable. Many reef fish also rely on moonlight for orientation and navigation during reproduction. In this sense, light acts as a universal signal that coordinates biological rhythms within the reef. Even the smallest changes in water transparency or temperature can shift these rhythms and alter the entire ecosystem's timing. Scientists studying coral reefs use advanced tools to measure how light behaves underwater. Instruments such as quantum sensors, spectroradiometers, and underwater cameras allow researchers to record the intensity and spectrum of light at different depths. Computer models then simulate how these light fields interact with coral geometry and pigmentation. Understanding these optical environments is crucial for predicting coral responses to environmental change. By modeling how much light reaches each layer of a reef, scientists can estimate photosynthetic productivity, calcification rates, and bleaching susceptibility. These data also help guide restoration efforts by identifying locations with optimal light conditions for coral transplantation. The energy produced by corals and algae does not stay within their partnership. It moves through the entire food web. Herbivorous fish feed on algae growing on dead coral surfaces, while plankton consume the organic compounds released by coral mucus. Predators, in turn, feed on these smaller organisms, creating a chain of energy that sustains the entire ecosystem. This integration between photosynthetic production and animal feeding makes coral reefs exceptionally efficient systems. They recycle nearly all available nutrients and maintain high productivity even in nutrient-poor tropical waters. Without light-driven photosynthesis, this balance would collapse. Light not only powers the reef, but also defines its limits. When light conditions remain stable, corals maintain their metabolic balance and support a complex web of life. When light is disrupted by warming, turbidity, or pollution, that balance is lost. Reefs that adapt or recover fastest are those capable of maintaining efficient photosynthesis under changing light regimes. This resilience depends on both biological diversity and environmental management. Protecting water clarity, reducing nutrient runoff, and minimizing temperature extremes help sustain the symphony of light that fuels coral ecosystems. Nightfall on the reef. When daylight fades, the coral reef enters a different phase of activity. Contrary to how it may appear during the day, the reef does not rest at night. Instead, it becomes one of the most dynamic environments in the ocean. As light diminishes, a complete shift occurs in behavior, feeding, and chemical exchange. The same structures that appeared quiet and still during the day transform into an intricate web of nocturnal life. Coral reefs operate on a daily rhythm governed by the cycle of light and darkness. During the day, energy production is dominated by photosynthesis. At night, when sunlight disappears, photosynthesis stops, and the reef community shifts from a phototrophic system to a heterotrophic one. This change affects nearly every organism living within the reef ecosystem. Coral polyps, which keep their tentacles retracted in daylight, begin to extend them once the light level drops. This is a feeding strategy. With the algae inside their tissues no longer producing sugars, the corals rely on capturing plankton and organic particles drifting in the water. The timing of this behavior aligns with the natural vertical migration of plankton, which rise toward the surface at night to feed. This synchronization allows corals to feed efficiently when prey density is highest. Each coral polyp has a simple structure: a mouth, a stomach cavity, and a ring of tentacles. At night, these tentacles expand into the surrounding water, increasing the coral's surface area for feeding. The stinging cells, or nematocysts, located on the tentacles, discharge upon contact with prey. Once captured, planktonic organisms are transported to the mouth, digested in the central cavity, and absorbed into the coral's tissues. Some corals produce mucus to trap food particles. The mucus film collects detritus and plankton from the water column, and then the coral reabsorbs it, obtaining both organic carbon and nitrogen. This feeding mechanism supplements the energy normally provided by photosynthesis, ensuring that corals have a stable energy supply across the 24-hour cycle. Feeding rates vary depending on the species and environmental conditions. In nutrient-rich or turbid environments, corals may rely more heavily on heterotrophic feeding than on photosynthesis. This flexibility allows some species to survive even in areas with low light or high sedimentation. As photosynthesis ceases after sunset, oxygen levels in reef waters begin to decline. During the day, algae and photosynthetic organisms release oxygen into the surrounding water. At night, all organisms, corals, fish, algae, and bacteria, consume oxygen through respiration. This can create strong diel fluctuations in oxygen concentration, especially in enclosed lagoons or areas with weak water movement. Measurements using oxygen sensors show that reef oxygen levels may drop sharply after dusk, sometimes becoming hypoxic in sheltered zones. To adapt, many organisms reduce their activity or move to areas with better circulation. Water currents become a critical factor in maintaining oxygen balance during the night. Carbon dioxide levels also rise during this period, lowering the pH of the surrounding water. This process, known as nighttime acidification, temporarily reduces the saturation state of calcium carbonate, slightly slowing coral calcification. However, corals compensate by increasing calcification again during daylight hours when photosynthesis resumes. The cycle of respiration and photosynthesis maintains long-term chemical equilibrium within the reef. When sunlight disappears, the community of active species within the reef changes dramatically. Many fish that rely on vision retreat into crevices or bury themselves in sand to avoid predators. As they hide, a different group of animals emerges. Crustaceans, such as shrimp and crabs, become more active at night, foraging for detritus and carrion. Sea urchins move across the reef surface, grazing on algae and biofilm. Polychaete worms and small snails crawl between coral branches, feeding on organic matter. Octopuses and moray eels leave their hiding spots to hunt, taking advantage of the reduced visibility. Predators on the night reef rely less on vision and more on chemical and pressure cues. They detect prey through vibrations, water movement, and chemical gradients. Some species of fish and crustaceans have highly sensitive lateral lines and antennae that allow them to locate prey even in near total darkness. The water above the reef becomes dense with plankton. Many of these organisms, including copepods, larval fish, and jellyfish, migrate upward each night from deeper layers. This phenomenon, known as diel vertical migration, is considered the largest synchronized movement of biomass on the planet. Corals and other filter feeders benefit directly from this event, capturing food that would otherwise be out of reach during the day. While the reef appears dark to human eyes, many marine organisms possess visual systems adapted to low light. Nocturnal fish often have large pupils and retinas dominated by rod cells, which are highly sensitive to dim light. Some species, such as squirrelfish and soldierfish, have reflective layers behind their retinas that enhance night vision. Bioluminescence is another common adaptation. Certain plankton, worms, and jellyfish produce light through chemical reactions within their bodies. This light can serve multiple functions: to attract prey, to communicate, or to confuse predators. When disturbed, bioluminescent plankton release flashes of light that ripple across the water, illuminating the outlines of swimming fish or passing currents. On nights with little moonlight, these bioluminescent flashes become one of the main sources of illumination on the reef. They are not decorative phenomena, but play measurable ecological roles, including predator avoidance and signaling between individuals. Some corals also exhibit fluorescence, caused by proteins within their tissues that absorb light of one wavelength and emit it at another. Under moonlight or artificial illumination, these corals appear to glow in green, orange, or red tones. This fluorescence is not true bioluminescence, as it does not require chemical reactions, but rather the re-emission of absorbed light. The function of coral fluorescence is still under study. It may help protect the coral from harmful radiation, enhance photosynthesis by redistributing light within the tissue, or play a role in communication and reproduction. Fluorescent proteins have also become valuable tools in biomedical research, as they can be used as natural markers to track molecular processes in cells. The transition from day to night also plays a central role in coral reproduction. Many coral species spawn only after sunset, releasing eggs and sperm into the water in synchronized events. This timing is controlled not just by daylight, but also by the lunar cycle. The brightness of moonlight, tidal currents, and water temperature combine to create predictable patterns that trigger mass spawning events. During such events, millions of gametes are released simultaneously, rising to the surface where fertilization occurs. This synchronization maximizes reproductive success by overwhelming predators and increasing the chances of cross-fertilization. In some regions, these mass spawning events occur only once a year, often just a few nights after the full moon. The precision of this timing illustrates the complexity of coral physiology. Light-sensitive proteins within coral cells detect changes in the lunar cycle, allowing corals to coordinate reproduction across vast distances. At night, when visibility is low, many reef organisms rely on chemical communication. Corals release compounds that can attract larvae or deter competitors. Some fish and crustaceans emit pheromones that help them locate mates or signal danger. Chemical cues also guide the settlement of coral larvae, helping them find suitable substrates for attachment. The concentration of these compounds fluctuates with time of day. Researchers have measured distinct chemical profiles in reef water during night hours, indicating that nocturnal activity changes the overall chemistry of the environment. This exchange of molecules creates an invisible network of communication linking all reef organisms. Calcification, the process by which corals build their skeletons, is influenced by the day-night cycle. During daylight, the process is enhanced by the high oxygen and pH levels created through photosynthesis. At night, as respiration dominates and pH decreases, calcification slows down but does not stop completely. Studies show that nighttime calcification rates may reach 20 to 40% of daytime levels. The reduced rate at night helps corals conserve energy. Instead of focusing on skeletal growth, they direct resources toward feeding, respiration, and repair. This balance between daytime growth and nighttime maintenance keeps coral colonies stable over long periods. Although it is often described as silent, the reef produces continuous sound, even in darkness. Nocturnal activity generates acoustic signatures detectable by hydrophones. The clicks of crustaceans, the grunts of fish, and the crackle of snapping shrimp combine into a constant background noise known as reef soundscape. These sounds play a biological role. They help larvae of fish and invertebrates locate reefs from kilometers away, guiding them to suitable habitats for settlement. At night, when visual cues are minimal, sound becomes a dominant form of environmental information. Nighttime on the reef is characterized by active predation and defense. Many nocturnal species are carnivorous, feeding on smaller invertebrates or juvenile fish. To survive, prey species adopt various defensive strategies. Some possess camouflage coloration that matches the surrounding coral. Others, like pufferfish, inflate their bodies when threatened. Certain crabs and shrimps use pieces of sponge or algae as physical shields. This constant interaction between predator and prey maintains population balance. The pressure of predation prevents any single species from becoming overly dominant, preserving biodiversity across the ecosystem. Studying coral reefs at night requires specialized equipment and techniques. Researchers use red light or low-intensity blue light to minimize disturbance. Diving at night allows observation of species that are hidden during the day, revealing behaviors that would otherwise go unnoticed. Infrared cameras, hydrophones, and oxygen sensors provide data on metabolic rates, movement, and sound. Through these studies, scientists have learned that more than half of reef species are primarily nocturnal. This includes many crustaceans, worms, and mollusks that form the base of the food web. Such findings highlight that daytime observations capture only part of the reef's true diversity. The nighttime period is crucial for redistributing energy within the reef ecosystem. Organic matter produced during the day by photosynthesis is consumed and decomposed by nocturnal organisms. This decomposition releases nutrients back into the water, making them available for algae and bacteria the following morning. The result is a continuous loop of production and recycling that maintains ecosystem stability. This cycle of transformation demonstrates that corals are not passive structures, but active participants in energy regulation. By alternating between photosynthesis and feeding, they keep the system balanced and resilient. The night phase of the coral reef is essential to its overall function. Without it, energy would accumulate unevenly, and essential nutrients would remain locked within organic matter. The activities of nocturnal species ensure that resources are constantly recycled. Predators keep prey populations in check. Scavengers and decomposers clean up waste. Filter feeders maintain water clarity. The combined effect is a complete ecosystem operating in two distinct but interconnected modes: a light-driven system during the day and a feeding-driven system at night. Together, these two phases sustain the extraordinary productivity and diversity of coral reefs, builders and breakers. Coral reefs are not static formations. They are dynamic systems built through continuous construction and destruction. The same biological processes that create reefs also contribute to their erosion. Every moment, even as new coral skeletons are deposited, others are being dissolved, broken, or consumed. This balance between growth and decay is what keeps a reef alive. A coral reef can be thought of as a living limestone factory. Billions of small organisms deposit calcium carbonate, forming hard structures that accumulate over centuries. At the same time, mechanical, biological, and chemical forces wear these structures down. The reef is therefore not a permanent monument, but a dynamic equilibrium, a balance between construction and deconstruction. If construction exceeds destruction, the reef expands. If destruction outpaces construction, the reef begins to erode and collapse. This competition between building and breaking defines the long-term health of any coral ecosystem. The foundation of reef building is calcification, the process by which corals and other organisms secrete calcium carbonate, KCO3, from seawater. In corals, this occurs beneath the living tissue layer, where ions of calcium and carbonate combine to form crystals of aragonite. These crystals fuse into a solid skeleton that supports the coral colony. Calcification is strongly influenced by light temperature, and water chemistry. During daylight, photosynthesis by symbiotic algae removes carbon dioxide from the coral's internal environment, raising pH and making it easier for calcium carbonate to form. This is why calcification rates are highest during the day. At night, as respiration dominates, pH decreases and the rate of skeleton formation slows. Other organisms also contribute to reef building. Calcareous algae, particularly crustose coralline algae, secrete calcium carbonate to form thin, hard layers that cover rocks and dead corals. These algae act as natural cement, binding loose fragments together and stabilizing the reef. Foraminifera, mollusks, and tube-building worms also deposit calcium carbonate, adding complexity and strength to the reef structure. This collective accumulation of calcium carbonate from multiple sources is known as biogenic accretion. It allows the reef to grow both vertically, following rising sea levels, and laterally, expanding into surrounding areas. While corals are the primary architects, many other organisms assist in constructing the reef's physical framework. Crustose coralline algae are particularly important. They grow over exposed surfaces, cementing coral fragments after storms and providing a stable foundation for new coral larvae to attach. Without these algae, reefs would be less stable and more prone to disintegration. Calcareous green algae, such as Halimeda, also contribute significantly. Their jointed, leaf-like structures are made of calcium carbonate plates. When these algae die, their remains break down into fine sand, which accumulates in reef lagoons and beaches. Giant clams and mollusks add to the carbonate budget through their shells, and tube worms construct small limestone tubes that further solidify the substrate. Collectively, these species form a network of bioconstructors that maintain reef growth even when coral cover fluctuates. Reef destruction, or bioerosion, is equally vital to the ecosystem's functioning. It may seem counterintuitive, but erosion allows reefs to recycle materials, create new habitats, and maintain structural balance. There are three main forms of reef erosion: mechanical, chemical, and biological. Mechanical erosion is caused by waves, storms, and currents. Strong waves break coral branches and grind them into smaller pieces. Over time, this mechanical action shapes the topography of the reef and produces sediment that forms beaches and islands. Chemical erosion occurs when seawater dissolves calcium carbonate, especially in conditions of low pH or high carbon dioxide concentration. This process has become more significant with ongoing ocean acidification. Biological erosion results from living organisms that bore, scrape, or consume coral skeletons. These organisms are collectively called bioeroders. Biological erosion is one of the most active and visible processes on a reef. It is carried out by organisms ranging from microscopic bacteria to large fish. Parrotfish are among the most effective bioeroders. They feed on algae that grow on coral surfaces, but in doing so, they bite off small pieces of the coral skeleton. The material they ingest is ground up in their digestive tract and excreted as fine sand. A single adult parrotfish can produce hundreds of kilograms of sand each year, contributing directly to the formation of tropical beaches. Sea urchins also play a major role. They use their specialized mouthparts, known as Aristotle's lantern, to scrape algae and thin layers of limestone from the reef surface. In moderate numbers, this grazing helps keep algae under control and prevents it from overgrowing corals. However, if urchin populations become too dense, their feeding can erode large portions of the reef structure. Boring sponges, worms, and bivalves, such as Lithophaga, bore into coral skeletons, creating tunnels and cavities. These weaken the reef's framework, but also provide habitat for other small organisms. In this way, Bioerosion contributes to both destruction and creation simultaneously. Microbial bioerosion, caused by bacteria and microalgae, operates on a microscopic scale. These organisms penetrate the calcium carbonate matrix, dissolving it chemically to obtain nutrients. Though invisible to the naked eye, their combined effect is substantial over long timescales. Tropical storms and hurricanes are natural parts of the reef's disturbance cycle. When strong waves strike the reef crest, they can break coral colonies into fragments or overturn large heads. Although this appears destructive, it also generates opportunities for regeneration. Broken coral pieces, if they settle in stable positions, can reattach to the substrate and continue growing. This process, known as fragmentation, is a natural form of asexual reproduction. In many reefs, post-storm regeneration contributes significantly to coral renewal. However, when storms occur too frequently or with extreme intensity, recovery time becomes insufficient. Repeated physical damage leads to long-term structural weakening and loss of habitat complexity. Scientists use the concept of a carbonate budget to measure the balance between reef construction and erosion. This budget quantifies how much calcium carbonate is produced by builders and how much is removed by breakers over a given period. A positive budget indicates that the reef is growing. A negative budget means that erosion exceeds growth, leading to net loss of structure. The carbonate budget is influenced by environmental factors such as temperature, nutrient levels, and ocean chemistry. Human impacts, including overfishing, pollution, and acidification, can shift the balance toward erosion. Monitoring the carbonate budget helps scientists predict the long-term stability of reef systems. The chemical stability of coral reefs depends on seawater pH. As atmospheric carbon dioxide increases, more CO2 dissolves into the ocean, forming carbonic acid. This lowers pH and reduces the availability of carbonate ions needed for calcification. Under such conditions, corals struggle to deposit skeletons, and existing calcium carbonate structures become more soluble. Acidification also enhances bioerosion by weakening coral skeletons, making it easier for boring organisms to penetrate. If pH continues to drop, the rate of chemical dissolution may exceed biological construction, causing reefs to gradually disintegrate. This process is slow but measurable, and represents one of the most serious long-term threats to coral ecosystems. Reef surfaces are constantly contested between corals and algae. When herbivorous fish and sea urchins graze regularly, they keep algae under control, allowing new corals to settle. When grazing pressure is reduced, for example through overfishing, algae can overgrow and smother living corals. This shift from coral to algal dominance is known as a phase shift, and once established, it can be difficult to reverse. Maintaining healthy populations of grazers is therefore critical for reef resilience. These organisms act as natural regulators, removing algae and dead tissue that would otherwise inhibit coral recruitment. Erosion and bioerosion are not purely destructive. They are essential for producing sediment that forms beaches, islands, and lagoons. The fine white sand found on many tropical shores originates mainly from ground coral skeletons and calcareous algae. Over thousands of years, this material accumulates, creating entire island chains such as the Maldives and Tuamotus. Thus, reef erosion contributes directly to land formation. Sediment transport and deposition also influence reef development. Too much sediment can smother living corals, but moderate sedimentation provides raw material for island growth and lagoon filling. This balance between production and deposition is part of the reef's natural geological cycle. Human activities have altered the balance between building and breaking in many reefs. Coastal development increases sediment runoff, while pollution adds nutrients that favor algal growth. Overfishing removes key grazers like parrotfish, reducing the reef's ability to control algae and maintain space for coral growth. In addition, global warming increases storm frequency and intensity, leading to more physical damage. The combination of these factors can push the carbonate budget into deficit. Reefs that once grew a few millimeters each year now lose mass faster than they can rebuild it. Scientific monitoring programs measure reef growth and erosion through underwater surveys, 3D mapping, and chemical analysis of water samples. These data show that in many parts of the world, reef growth has slowed by more than half in the past few decades. Despite the damage it can cause, erosion has ecological value. By breaking down old structures, it creates new habitats for colonization. Small holes, cracks, and crevices formed by bioeroders provide shelter for invertebrates, fish larvae, and microorganisms. This microhabitat diversity increases overall biodiversity and productivity. Reef systems rely on disturbance as much as on stability. Without occasional breakdown, coral surfaces would become smooth and uniform, reducing habitat complexity. Erosion therefore serves as a form of ecosystem renewal, clearing space for new growth and maintaining structural heterogeneity. Over geological time, the interplay between construction and erosion determines whether reefs survive environmental changes. During periods of rising sea level, active reefs grow upward to stay within the photic zone. If they cannot keep pace, they drown and become fossilized beneath new sediment layers. Conversely, during sea level falls, exposed reefs undergo chemical dissolution and mechanical weathering. But parts of them may later be uplifted by tectonic forces to form limestone cliffs and islands. Fossil reefs found on land record millions of years of such cycles. They provide evidence of past climates, sea level fluctuations, and ocean chemistry. By studying these ancient structures, scientists gain insight into how modern reefs might respond to future change. The living reef is therefore not a fixed object, but a dynamic process, a continuous interaction between construction and destruction. Every piece of coral rock on the sea floor once represented a living organism. Every grain of sand was part of a coral skeleton or an algal plate. The reef grows, erodes, and rebuilds itself in a constant cycle of transformation. This balance allows reefs to adapt and persist over long timescales. They are resilient precisely because they incorporate both creation and decay into their structure. Understanding this dynamic is essential for conservation. Protecting reefs does not mean freezing them in a single state, but ensuring that the natural processes of building and breaking can continue within healthy limits. The Fragile Kingdom. Coral reefs are among the most productive and diverse ecosystems on Earth, but they are also among the most fragile. The conditions that allow corals to grow, warm, clear, stable, and nutrient-poor waters, are precisely the conditions most easily disrupted by environmental change. Because of this sensitivity, coral reefs act as early indicators of stress in the ocean. When the balance shifts, they respond quickly and often dramatically. Reef-building corals live within a narrow environmental window. They require sunlight for photosynthesis, temperatures typically between 23 and 29 degrees Celsius, and stable salinity around 35 parts per thousand. They also depend on clear water with low sediment and nutrient levels. When these conditions are stable, coral growth and reproduction proceed efficiently. When they fluctuate beyond natural limits, even slightly, the biological systems that sustain the reef begin to fail. This sensitivity comes from the coral's physiology. Its relationship with symbiotic algae depends on chemical equilibrium inside the coral tissue. Any disturbance that alters temperature, light, or pH affects that equilibrium, disrupting the partnership and reducing energy production. As a result, environmental changes that might be tolerable to other marine organisms can be fatal to corals. One of the most well-known threats to coral reefs is thermal stress, which leads to a phenomenon known as coral bleaching. When water temperatures rise just 1 or 2 degrees Celsius above the average summer maximum, the photosynthetic machinery of the symbiotic algae begins to malfunction. This malfunction causes the production of reactive oxygen molecules, which are toxic to both the algae and the coral host. To protect itself, the coral expels the algae from its tissues. Without algae, the coral loses its main source of food and color, leaving behind a white skeleton visible through its transparent tissue, hence the term bleaching. If temperatures return to normal within a few weeks, the coral can reabsorb algae and recover. However, prolonged heat stress leads to starvation, disease, and death. Mass bleaching events have become more frequent since the 1980s, corresponding with global increases in sea surface temperatures. In some regions, entire reef systems have experienced bleaching at scales once thought impossible. The Great Barrier Reef, for example, has suffered multiple widespread bleaching events since 2016, affecting over half of its corals. Scientists use the metric degree heating weeks, DHW, to measure accumulated heat stress over time. A DHW value above 4 indicates likely bleaching, while values above 8 often result in severe or widespread mortality. These metrics allow researchers to predict and map bleaching risk in near real time using satellite data. In addition to warming, ocean acidification poses another long-term threat. As atmospheric carbon dioxide increases, a significant portion dissolves into seawater, forming carbonic acid. This lowers the ocean's pH and decreases the concentration of carbonate ions. The building blocks corals need to produce calcium carbonate. The result is slower calcification and weaker skeletons. Acidification also increases the solubility of existing coral skeletons, making them more prone to chemical erosion. Laboratory studies show that even small decreases in pH can reduce coral growth rates by 20 to 50%. If acidification continues, many reefs will lose their structural integrity, collapsing faster than they can rebuild. Unlike bleaching, which is visible and often sudden, acidification is subtle and progressive. It affects not only corals, but also other calcifying organisms such as mollusks, echinoderms, and plankton. This global process alters the ocean's chemistry in ways that will persist for centuries. Pollution is another major driver of coral reef degradation. Runoff from agriculture, sewage, and industry introduces nutrients, chemicals, and sediments into coastal waters. Excess nutrients, particularly nitrogen and phosphorus, promote the growth of algae. These algae can overgrow coral surfaces, blocking light and smothering polyps. Fine sediments have similar effects. They settle on coral tissues, clogging feeding structures and reducing photosynthetic efficiency. To remove these sediments, corals produce mucus, which requires significant energy. If the sedimentation continues, the corals eventually suffocate. Chemical pollutants, including heavy metals, oil residues, and pesticides, can interfere with coral reproduction and immune responses. Even trace amounts of some sunscreen ingredients, such as oxybenzone and octinoxate, have been shown to cause deformities in coral larvae and inhibit settlement. Several countries have already banned such chemicals to protect their reefs. Coral reefs depend on complex food webs to maintain balance. Overfishing disrupts these relationships. When herbivorous fish like parrotfish and surgeonfish are removed, algae grow unchecked, covering the substrate and preventing coral recruitment. The loss of predators, such as groupers and snappers, can also lead to population explosions of smaller species that feed directly on coral tissue or larvae. Destructive fishing methods, including blast fishing and cyanide fishing, have caused extensive physical damage to reefs in parts of Southeast Asia and the Pacific. These practices not only destroy coral structures instantly, but also reduce the reef's ability to regenerate. Fish populations recover slowly, and once algal dominance sets in, the entire ecosystem may shift to a less diverse and less productive state. This process can occur within just a few years of heavy exploitation. Human construction near coastlines, harbors, resorts, breakwaters, and dredging has direct and indirect effects on reefs. Dredging increases turbidity, reducing the amount of light reaching the reef. Land reclamation changes water circulation patterns, leading to sediment accumulation in new areas. Physical contact from anchors, boats, and divers causes breakage and abrasion of fragile coral colonies. In heavily developed regions, these local impacts often combine with global stressors such as warming and acidification. Together, they create conditions that exceed the coral's ability to adapt. In recent decades, coral diseases have become increasingly common. These diseases are caused by bacteria, fungi, and viruses, and their outbreaks are often linked to environmental stress. Examples include black band disease, white band disease, and stony coral tissue loss disease, SCTLD. Black band disease forms a dark, microbial mat that moves slowly across coral surfaces, killing tissue as it progresses. White band disease affects branching corals, stripping away living tissue and leaving white skeletons behind. SCTLD, first identified in the Caribbean, spreads rapidly and affects dozens of coral species, causing tissue necrosis and high mortality rates. High water temperatures and poor water quality increase the likelihood of infection. When corals are stressed, their immune systems weaken, and pathogens that are normally harmless can become virulent. Unlike many terrestrial diseases, treating or containing coral diseases in the ocean is extremely difficult. Climate change affects coral reefs not only through gradual warming, but also through changes in weather patterns and storm frequency. Tropical cyclones, hurricanes, and typhoons have become more intense in many regions. Powerful waves break coral structures, dislodge colonies, and generate debris fields. After severe storms, reef recovery can take decades. The degree of recovery depends on whether surviving corals can reproduce and reattach. In areas where additional stressors exist, such as high sedimentation or overfishing, the likelihood of full recovery is much lower. Sea level rise adds further complications. While reefs can grow vertically to keep pace with rising water, they require stable conditions and sufficient carbonate production. If growth rates slow due to warming or acidification, reefs may drown beneath the surface, losing their function as wave barriers and habitats. Invasive species can also disrupt reef ecosystems. The most famous example is the crown-of-thorns starfish, Acanthaster planchi. This coral-eating species naturally occurs on many reefs, but under certain conditions, such as increased nutrient availability, its population can explode. Each individual starfish can consume several square meters of coral per year. Outbreaks can devastate large reef areas, leaving behind barren surfaces. Other invasive species include algae, sponges, and even fish introduced through shipping or aquaculture. Once established, they compete with native species for space and resources, further stressing the reef ecosystem. In reality, coral reefs rarely suffer from a single disturbance. They experience multiple stressors acting simultaneously or sequentially. Warming, acidification, pollution, and overfishing interact in complex ways. For example, heat stress may make corals more susceptible to disease, while nutrient pollution can intensify algal growth after bleaching events. The combined effect is often greater than the sum of individual impacts. This phenomenon is known as cumulative stress. It reduces the reef's resilience, making it harder for corals to recover between disturbances. The frequency of extreme events, heat waves, storms, disease outbreaks, now exceeds the natural recovery time of many reefs. Despite their vulnerability, coral reefs are capable of recovery under favorable conditions. Recovery begins with the settlement of coral larvae on stable surfaces. If water quality improves and grazing fish return, algae are kept under control, and new coral colonies can grow. This process may take several years to decades, depending on species and location. Some corals exhibit adaptive bleaching responses, in which they acquire more heat-tolerant algae after a bleaching event. This change can improve their resilience to future temperature increases. Genetic variation within coral populations also plays a role. Certain individuals or species naturally tolerate higher temperatures or acidity, and their offspring inherit these traits. This gradual adaptation may help some reefs persist even as environmental conditions shift. Humans are now actively trying to aid reef recovery through restoration. Techniques include coral gardening, where fragments are grown in nurseries and later transplanted onto damaged reefs. Microfragmentation accelerates growth by cutting large corals into small pieces, which then fuse together more quickly as they heal. Other methods involve artificial substrates made of metal, concrete, or biodegradable materials to provide stable surfaces for coral attachment. Some projects experiment with biorock technology, using low electric currents to stimulate calcium carbonate deposition. Others explore selective breeding and genetic research to identify resilient coral strains. While these efforts show promise on local scales, they cannot yet compensate for global threats such as warming and acidification. Successful restoration depends on addressing root causes, reducing greenhouse gas emissions, controlling pollution, and managing fisheries sustainably. The loss of coral reefs carries significant human consequences. Reefs provide coastal protection, food security, and livelihoods for hundreds of millions of people. They attract tourism, support fisheries, and maintain biodiversity. When reefs degrade, these benefits disappear. In small island nations, coral loss directly threatens economic stability and safety. Without reefs, waves erode shorelines, destroy infrastructure, and contaminate freshwater supplies. Globally, the economic value of coral reef ecosystem services is estimated at hundreds of billions of dollars annually. Protecting reefs is therefore not just an ecological issue, but also a socioeconomic necessity. Ecologists warn that coral reefs may reach tipping points, thresholds beyond which recovery becomes impossible. Once coral cover falls below a certain level, the system can shift permanently from coral-dominated to algae-dominated. This new state supports fewer species and provides fewer ecosystem services. Reversing it would require removing multiple stressors simultaneously, a task rarely achieved. Understanding these tipping points is essential for conservation planning. By identifying early warning indicators, such as reduced calcification, increased disease, or shifts in fish populations, scientists can act before irreversible changes occur. The fragility of coral reefs is not a sign of weakness, but of precision. They exist in balance with their surroundings, responding to small changes with sensitivity that reflects their fine-tuned adaptation. Protecting them requires maintaining that balance, not only by preventing destruction, but also by allowing natural processes of growth and renewal to continue. Preserving water quality, reducing global emissions, and managing coastal development responsibly are all part of the same goal. Keeping the physical and biological conditions within the narrow limits that allow corals to thrive. The survival of coral reefs depends on both local and global action. Their decline is not inevitable, but recovery will require long-term commitment, informed management, and respect for the limits of these extraordinary ecosystems. Love and life of coral. The life cycle of coral is one of the most remarkable and complex processes in marine biology. Although coral colonies may live for centuries, their survival as a species depends on brief, highly coordinated events that occur only a few times each year. These reproductive cycles determine how reefs expand, recover, and adapt to changing conditions. Understanding them reveals how coral populations maintain genetic diversity and resilience over long timescales. Corals reproduce both asexually and sexually, using different strategies to ensure survival. Asexual reproduction allows colonies to grow and repair themselves. Sexual reproduction introduces genetic variation, which is essential for adaptation to environmental change. Asexual reproduction occurs through budding or fragmentation. In budding, a new polyp forms from an existing one and remains attached, expanding the colony. This process enables continuous growth and helps the coral maintain structural integrity after physical damage. Fragmentation happens when a piece of coral breaks off, often due to waves or storms, and reattaches to a new substrate. If the fragment settles in a stable position, it can grow into a new colony genetically identical to the parent. This ability to regenerate gives corals a natural capacity for recovery after disturbances. Sexual reproduction, however, is what connects different colonies and sustains the genetic diversity of the species. Most reef-building corals are broadcast spawners, releasing eggs and sperm into the water column. A smaller number are brooders, where fertilization occurs internally and larvae are released later. Each method has advantages suited to different environments. Broadcast spawning is the dominant reproductive strategy among corals. It involves the simultaneous release of eggs and sperm by many colonies across wide areas of the reef. The timing of spawning is highly synchronized, often occurring on a few specific nights each year. The synchronization depends on several environmental cues: water temperature, daylight length, tidal patterns, and lunar cycles. Most species spawn shortly after sunset, a few days after a full moon, when tidal currents are calm and conditions favor fertilization. The event can be so extensive that the sea surface appears covered with pink or orange slicks of coral gametes. Fertilization occurs externally when eggs and sperm meet in the water column. The resulting embryos develop into planula larvae, tiny, free-swimming organisms that drift with currents for days or weeks before settling. This planktonic stage allows genetic exchange between distant reefs, maintaining connectivity across large regions. However, broadcast spawning is a high-risk strategy. Only a small fraction of gametes survive predation, currents, or unfavorable conditions. The success of each event depends on precise timing, healthy parent colonies, and suitable environmental conditions. Some coral species follow a different approach, known as brooding. In these corals, fertilization takes place inside the polyp. The larvae develop within the parent colony and are released when fully formed. This strategy reduces losses from predation and dispersal. Brooded larvae are generally larger and contain symbiotic algae from birth. which gives them an immediate energy source after settlement. However, their dispersal range is shorter, making them more dependent on local conditions and limiting genetic mixing. Brooding is therefore common in environments where stability and substrate availability are high. After fertilization, the coral embryo develops into a planula, a small, oval-shaped larva covered in cilia, tiny hair-like structures used for swimming. The planula stage typically lasts from a few days to several weeks. During this time, larvae are carried by ocean currents, drifting across potentially vast distances. Larvae are sensitive to light, gravity, and chemical cues. They tend to migrate toward the surface during the day and sink at night, a behavior that helps them stay within optimal temperature and salinity ranges. Eventually, when they encounter a suitable surface, they attach and transform into the first polyp of a new colony, a process known as settlement and metamorphosis. Settlement preferences are highly specific. Larvae often choose surfaces coated with crustose coralline algae, which emit chemical signals promoting attachment. Clean, hard substrates are preferred, while surfaces covered by sediment, algae, or sponges are avoided. This selectivity ensures that new corals begin life in areas favorable for survival. Once a larva attaches, it secretes calcium carbonate to form its initial skeleton, establishing the foundation of the new colony. The young polyp then begins to feed, extend tentacles, and divide through budding. Within weeks, a small colony forms, consisting of genetically identical individuals connected by living tissue. Early growth is a critical stage in coral development. Juvenile corals are vulnerable to competition, predation, and physical disturbances. Many are grazed by fish or overgrown by algae. Survival rates during the first year are often below 10%. Those that survive grow slowly but steadily, eventually becoming large enough to withstand minor disturbances and participate in reproduction themselves. Coral colonies exhibit various growth forms depending on species and environmental conditions. Branching corals, such as Acropora, grow quickly, extending several centimeters per year, but they are fragile and prone to breakage. Massive corals, such as porites, grow only a few millimeters per year, yet can live for hundreds of years. Their solid, dome-shaped skeletons resist waves and erosion. Growth is influenced by light availability, temperature, water movement, and sedimentation. Corals on upper reef slopes, exposed to strong currents, often develop compact forms to minimize physical stress. In sheltered areas, flatter and broader shapes dominate. These growth patterns contribute to the structural complexity that defines a healthy reef. Most corals reach sexual maturity within 3 to 8 years, depending on the species. Once mature, colonies may spawn annually or several times per year in equatorial regions. Spawning is energy intensive. In the months leading up to the event, corals accumulate lipids and proteins to support gamete production. Environmental cues, especially temperature and lunar cycles, synchronize the release of gametes within populations. This synchronization ensures that eggs and sperm from different colonies meet in the water, maximizing fertilization success. Even minor shifts in temperature or moon phase can alter spawning timing, potentially reducing reproductive efficiency. Sexual reproduction allows corals to exchange genetic material across reefs. Ocean currents transport larvae between distant populations, promoting gene flow and maintaining diversity. Genetic diversity enhances resilience by providing a range of traits that may confer tolerance to heat, acidity, or disease. However, fragmentation of reefs due to human activities can reduce connectivity. When isolated, populations become more vulnerable to local extinctions. Maintaining pathways for larval dispersal, sometimes called larval corridors, is therefore a priority for marine conservation planning. In recent years, scientists have discovered that corals may adapt faster than previously thought. Through a process called symbiont shuffling, they can change the composition of their internal algae, replacing sensitive types with more heat-tolerant ones. This flexibility allows short-term adaptation to environmental stress. Beyond reproduction, corals have impressive regenerative capabilities. When a colony is damaged by storms, fish, or human activity, the remaining tissue can regrow over exposed skeletons. Some corals can regenerate entire branches from small surviving fragments. This process plays a crucial role in reef recovery following physical disturbances. Experiments show that certain species can repair partial tissue loss within days. Others require months, depending on energy reserves and environmental conditions. Regeneration is energy demanding, and prolonged stress or poor water quality can slow or prevent recovery. Reproductive success in corals depends heavily on environmental stability. Temperature fluctuations can delay or inhibit spawning. High turbidity reduces fertilization rates by dispersing gametes and blocking chemical cues. Pollution and sedimentation can damage gametes and larvae before settlement. Chemical pollutants, particularly hydrocarbons and heavy metals, interfere with coral endocrine systems, reducing reproductive output. Light and lunar cycles are especially critical. Some corals rely on specific light wavelengths to trigger gamete release. Even artificial lighting from coastal development can disrupt these cues, leading to desynchronized spawning and reduced reproductive success. Coral reproduction involves significant trade-offs between growth, maintenance, and reproduction. Allocating energy to gamete production can temporarily reduce calcification and tissue repair. Species in stable environments tend to invest more in sexual reproduction, while those in disturbed areas rely more on asexual strategies. Studies show that reproductive output often decreases after bleaching events or prolonged stress. Recovery of energy reserves may take several years, delaying future spawning cycles and slowing population recovery. Settlement success depends on both larval behavior and habitat condition. Healthy reefs with abundant crustose coralline algae and clean substrates attract more settlers. Damaged reefs covered by sediment or macroalgae repel larvae chemically. This creates a feedback loop. Degraded reefs attract fewer recruits, leading to further decline, while healthy reefs reinforce their own regeneration. Experiments with artificial settlement tiles and acoustic cues show that larvae can detect reef sounds and chemical signals from thriving ecosystems. They actively choose environments that indicate stability and productivity. This behavioral selectivity enhances survival rates of new generations. Mass spawning events represent a remarkable example of biological coordination. By synchronizing reproduction, corals maximize genetic exchange and population renewal. These events also serve as critical food sources for many reef organisms. Fish, crustaceans, and plankton feed on coral gametes, integrating reproductive energy into the wider ecosystem. Although most gametes do not become new corals, they contribute to the nutrient dynamics that sustain the reef as a whole. Such synchronization depends on stable environmental signals. Disruptions caused by climate change, altered currents, irregular moon phases, or temperature anomalies may desynchronize these cycles, reducing reproductive efficiency across entire reef systems. Some coral colonies, particularly massive ones, can live for hundreds or even thousands of years. Radiometric dating of coral skeletons reveals ages exceeding 4 millennia in certain species. These long-lived organisms act as archives of environmental history, recording temperature, salinity, and nutrient changes in their skeletons. Despite this longevity, individual polyps within a colony live only a few years, continually replaced by new ones through budding. The colony as a whole persists, passing its genetic material across countless generations of polyps. This modular form of life blurs the line between individual and collective existence. The reproduction and regeneration of corals demonstrate the resilience of life in extreme environments. Every year, across thousands of reefs, millions of eggs and larvae begin their journey in the open ocean. Most will not survive, but a small fraction will find a place to attach, grow, and eventually build new structures of stone and color. This continuous cycle of loss and renewal maintains coral populations even in unstable conditions. It is a process shaped by both chance and adaptation, where survival depends on timing, environment, and cooperation between species. Through these reproductive strategies, sexual, asexual, and symbiotic, corals persist as one of the oldest and most successful groups of animals on Earth. The hidden voices. To the human ear, a coral reef seems silent. When divers descend beneath the surface, the first sensation is stillness, a world without speech, without language. But scientific instruments reveal a very different reality. Reefs are full of sound, signal, and chemical conversation. Every species, from the smallest bacterium to the largest fish, participates in an invisible network of communication that coordinates life beneath the waves. These exchanges take many forms: mechanical vibrations, acoustic emissions, chemical gradients, and even microbial signals at the cellular level. Together, they form what scientists call the communication ecology of the reef, a continuous flow of information that guides movement, reproduction, feeding, and cooperation. The term soundscape describes the collective acoustic environment of an ecosystem. In coral reefs, this soundscape is surprisingly complex. Hydrophones placed near healthy reefs record constant background noise, a mixture of crackles, pops, clicks, and low hums. Most of these sounds are produced by snapping shrimp, fish vocalizations, and other invertebrates. Snapping shrimp, for instance, create sharp popping sounds by closing their claws so fast that they generate tiny bubbles, which collapse and produce intense pressure waves. A dense population of snapping shrimp can generate a continuous crackling noise, audible from hundreds of meters away. Fish also contribute to the soundscape. Many reef fish species produce sounds to attract mates, defend territory, or coordinate group behavior. They generate these noises by vibrating their swim bladders or grinding their teeth. For example, damselfish emit short bursts during aggressive encounters, while toadfish produce low-frequency hums during mating. The combined result is a constant background signal, a kind of underwater static that indicates biological activity. When a reef is healthy and full of life, this sound is strong and continuous. When a reef is degraded or dead, the soundscape becomes quiet. Thus, sound can serve as a diagnostic indicator of reef health. For many marine species, sound is more than noise. It is a navigational tool. Fish larvae and coral planulae drifting in the open ocean use the reef's soundscape to locate suitable habitats. Laboratory experiments have shown that larvae actively swim toward reef sounds and avoid silent areas. This ability to detect and respond to sound is called phonotaxis. It allows larvae to return to reefs after dispersal, maintaining population connectivity. Sound travels 4 to 5 times faster in water than in air, and low-frequency vibrations can carry long distances. A healthy reef can therefore attract new life, even from kilometers away. Artificial sound playback experiments confirm this effect. When speakers broadcasting reef noises are placed near degraded areas, larvae settle more frequently, leading to higher recruitment rates. Such studies suggest that restoring the acoustic environment could become a useful tool in reef rehabilitation. The reef's acoustic profile changes over time. During the day, sounds are dominated by fish and invertebrates that feed actively under light. At night, many fish become silent, while nocturnal crustaceans and worms take over. These diel cycles of sound reflect behavioral rhythms of reef inhabitants. Seasonal variations also occur. During breeding periods, the intensity of fish calls increases significantly, creating what researchers call choruses. These collective signals can last for hours and are associated with reproductive synchronization. Recording long-term changes in soundscapes helps scientists track ecosystem shifts. A gradual decline in sound intensity often indicates reduced biodiversity or coral cover, even before visual degradation becomes apparent. Not all reef communication occurs through airborne-like sound waves. Many species use substrate-borne vibrations, mechanical signals transmitted through rock or sand. Certain fish, crabs, and shrimps detect these vibrations using specialized sensory organs. For example, hermit crabs can sense approaching predators through minute vibrations in the substrate, allowing them to retreat before danger becomes visible. Corals themselves respond to vibration and water movement. Although they lack nerves and ears, they possess mechanoreceptor-like cells capable of detecting changes in pressure and flow. Laboratory experiments show that polyp expansion and tentacle orientation can change in response to vibrations caused by nearby swimming fish. This sensitivity helps corals optimize feeding and avoid mechanical damage. Beyond sound and vibration, the primary medium of communication on the reef is chemistry. The ocean is a liquid matrix rich in dissolved molecules. Every organism releases compounds, metabolic waste, pheromones, defensive toxins, and stress signals that diffuse through the water and carry information to others. Corals release dissolved organic matter and mucus, which not only feed microorganisms, but also influence the behavior of nearby species. Some of these compounds attract fish and invertebrates that clean or protect the coral. Others deter predators or inhibit the growth of competing organisms. Fish use chemical cues to locate spawning grounds or identify kin. Larvae detect specific scents from healthy reefs or particular coral species that guide them toward appropriate habitats. Even microorganisms respond to chemical gradients, moving toward or away from certain compounds in a process known as chemotaxis. Chemical signals also play a role in coral competition. Neighboring colonies of different species sometimes engage in chemical warfare. They release allelopathic substances that inhibit each other's growth or kill adjacent tissue. This chemical conflict shapes the spatial distribution of coral species across the reef. Coral health depends heavily on its microscopic partners. A coral colony is not just a single organism, but a holobiont, a consortium of the coral animal, symbiotic algae, bacteria, archaea, fungi, and viruses. These microorganisms perform essential functions such as nutrient cycling, nitrogen fixation, and disease resistance. Microbes communicate through chemical signaling known as quorum sensing. They release molecules that allow them to coordinate activities such as biofilm formation or defense response. When the microbial community is balanced, these interactions maintain coral health by regulating immune responses and preventing pathogen invasion. When disrupted by stress or pollution, harmful bacteria can dominate, leading to disease. Researchers are increasingly recognizing the coral microbiome as a dynamic and adaptive system. Some bacteria may help corals tolerate higher temperatures or resist infection. Manipulating these microbial communities sometimes called probiotic intervention, is an emerging area of reef restoration research. Chemical cues are also essential for coral reproduction. When coral larvae search for settlement sites, they rely on specific molecules released by crustose coralline algae and microbial biofilms. These cues trigger metamorphosis from free-swimming larvae to attached polyps. If the chemical signals are absent due to pollution, sedimentation, or algal overgrowth, larvae may fail to settle, drifting until they die. The presence of healthy microbial films and crustose algae thus directly influences reef renewal. These subtle interactions between microorganisms and coral larvae determine where new generations begin. Corals and their symbiotic algae also exchange biochemical signals in response to light. Changes in light intensity or spectrum alter the production of photosynthetic pigments and proteins. Corals produce fluorescent and chromoproteins that regulate light absorption and protect tissues from ultraviolet radiation. These pigments may also act as internal communication molecules, signaling oxidative stress between cells. At the biochemical level, coral cells and symbionts exchange reactive oxygen species and antioxidant compounds. This ongoing dialogue helps maintain homeostasis under fluctuating light and temperature. When the communication fails, as during heat stress, the breakdown leads to bleaching. When corals are injured or stressed, they release chemical alarm compounds into the surrounding water. These substances can trigger defensive responses in neighboring colonies. For example, nearby corals may retract their polyps, increase mucus production, or activate antimicrobial defenses. This phenomenon, known as allelobiosis, shows that coral colonies can influence each other's behavior chemically. Similarly, some fish and invertebrates respond to alarm cues from conspecifics. When one individual is attacked, others detect released molecules and hide or flee. This shared signaling network reduces overall predation risk. Communication among microorganisms can also determine disease outcomes. Pathogenic bacteria often use quorum sensing to coordinate infection. They produce toxins only when their population density is high enough to overwhelm the host's defenses. Blocking these communication signals can prevent disease without killing the bacteria directly, a strategy currently being explored for reef protection. Environmental stress can disrupt microbial balance, allowing opportunistic pathogens to proliferate. Understanding these microbial communication networks helps scientists predict disease outbreaks and develop targeted mitigation approaches. The reef has a distinct chemical smell that varies between healthy and degraded states. Healthy reefs release a mixture of volatile organic compounds derived from algae, bacteria, and coral mucus. These compounds attract fish larvae and other organisms seeking settlement sites. When a reef becomes overgrown with algae or affected by decay, its chemical profile changes dramatically. The altered scent can repel larvae and reduce recruitment, accelerating ecological decline. This discovery highlights that coral conservation is not only about visual structure, but also about maintaining the invisible chemical environment that sustains life. All these communication systems, acoustic, vibrational, chemical, and microbial, are interconnected. For example, increased sound activity often corresponds with high chemical productivity. Dense microbial communities influence the reef's chemical profile, which in turn affects larval settlement and acoustic behavior of fish. These feedback loops create a self-regulating network of biological information. When the network is intact, the reef remains stable and resilient. When it is disrupted by noise pollution, chemical contamination, or biodiversity loss, the flow of information weakens, and ecological coordination breaks down. Human-generated noise increasingly interferes with natural reef soundscapes. Ship engines, construction, and sonar produce low-frequency sounds that can mask biological signals or disturb marine life. Chronic noise exposure can disrupt fish communication, alter migration patterns, and reduce larval settlement. Studies show that reefs near busy shipping lanes are significantly quieter biologically, reflecting lower species abundance and activity. Managing acoustic pollution is now considered part of marine conservation. Reducing ship speeds, rerouting traffic, and setting quiet zones help maintain natural sound conditions that support reef function. Chemical contamination also interferes with reef communication. Pesticides, hydrocarbons, and heavy metals can alter the production or perception of natural chemical cues. Even low concentrations may block the chemical signals that guide larval settlement or symbiotic interactions. Some pollutants mimic natural signaling molecules, creating confusion within the ecosystem. This phenomenon, called chemical noise, makes it harder for organisms to distinguish meaningful cues from background contamination. Through advanced technologies, hydrophones, chemical sensors, and genetic sequencing, scientists are now decoding these hidden voices of the reef. Each type of signal offers clues about ecological health. A vibrant soundscape indicates active biodiversity. Stable chemical profiles reflect balanced microbial activity. Disruptions in either often precede visible coral decline. By listening and measuring, rather than only observing, researchers gain a more complete understanding of how reefs function as living communication networks. These insights are transforming coral science from descriptive biology into systems ecology, where sound, chemistry, and microbiology merge into one dynamic picture, healing the reef. Coral reefs, though fragile, possess an inherent ability to heal. Their survival over hundreds of millions of years attests to this resilience. They have endured mass extinctions, sea level changes, and climate fluctuations. But the speed and intensity of modern environmental change have pushed that resilience to its limits. Understanding how reefs recover and how humans can support that recovery is central to the future of tropical oceans. Reef healing occurs on multiple scales, from the microscopic regeneration of coral tissue to the large-scale restoration of entire ecosystems. It combines natural biological processes with human intervention and management. At its core, healing depends on time, stability, and balance, the same conditions that allowed reefs to form in the first place. Coral reefs possess intrinsic repair systems. When physical damage occurs, such as broken branches or partial tissue loss, corals initiate a healing process similar to wound closure in other animals. Cells around the injured area proliferate and migrate to cover exposed skeletons. Over days or weeks, new tissue forms, sealing the damaged surface. If conditions remain stable, calcification resumes, and the coral slowly regains its shape. Some corals rely on fragmentation as a natural form of regeneration. When branches break off during storms, the fragments may reattach to stable substrates, continuing to grow as independent colonies. This form of asexual propagation is a key survival mechanism, allowing rapid recolonization after disturbance. Reefs also recover through larval recruitment, the settlement of new coral larvae produced by sexual reproduction. Healthy reefs with abundant parent colonies can repopulate damaged areas within a few years, provided that water quality and grazing activity remain favorable. For recovery to occur, several environmental factors must align. Clear water allows light penetration, supporting photosynthesis by symbiotic algae. Stable temperatures prevent bleaching and promote tissue repair. Balanced nutrient levels discourage excessive algal growth, while healthy populations of herbivorous fish control algae naturally. Connectivity between reefs is also critical. Larvae must be able to drift from healthy reefs to degraded ones. Ocean currents serve as biological highways, transporting genetic material across vast distances. If connectivity is lost due to habitat fragmentation or pollution, damaged reefs remain isolated and struggle to recover. Certain species act as natural restorers of reef balance. Herbivorous fish, such as parrotfish and surgeonfish, prevent algae from overgrowing coral surfaces. Sea urchins also contribute by scraping rocks clean, creating space for coral larvae to settle. When these grazers are overfished or removed, algae can dominate, forming a barrier to coral recovery. Restoring their populations often leads to visible ecological improvement. In several marine reserves, banning fishing for key herbivores has resulted in coral cover doubling within a decade. Predators, though fewer in number, also maintain ecological stability. By keeping smaller fish populations in check, they prevent overgrazing of critical algae and maintain food web balance. One of the most effective tools for promoting natural reef recovery is the establishment of marine protected areas. MPAs restrict or regulate human activities such as fishing, mining, and tourism. By reducing stress, they allow ecosystems to rebuild naturally. Long-term studies show that reefs within MPAs have higher coral cover, greater biodiversity, and stronger fish populations than those in unprotected zones. MPAs act as reservoirs of genetic diversity, supplying larvae to neighboring degraded reefs through larval dispersal. However, protection alone is not sufficient. MPAs must be properly enforced and integrated into broader coastal management strategies. Their success depends on community participation, economic alternatives for local fishers, and continuous scientific monitoring. When natural recovery is too slow, scientists and conservationists intervene directly through active restoration. One of the most widely used methods is coral gardening. This approach involves collecting small coral fragments from healthy colonies and growing them in underwater nurseries suspended in mid-water or attached to artificial structures. Once the fragments reach sufficient size, they are transplanted onto degraded reefs using cement, epoxy, or cable ties. These fragments continue to grow, gradually forming new colonies. Over time, they can merge, restoring coral cover and habitat complexity. Coral gardening has been implemented successfully in the Caribbean, the Philippines, Indonesia, and the Maldives. Survival rates vary by species and site conditions. But under optimal management, more than 80% of transplanted corals can survive their first year. Recent advances have refined restoration techniques further. Microfragmentation involves cutting corals into very small pieces, as small as 1 square centimeter. These microfragments grow and fuse rapidly, accelerating the recovery of massive coral species that normally grow very slowly. When placed close together on artificial substrates, the fragments recognize each other as genetically identical and merge to form larger colonies within months. Another innovation is assisted evolution, in which corals are selectively bred or exposed to controlled stress in laboratories to enhance their tolerance to heat and acidification. Researchers then reintroduce these resilient strains into natural reefs. Although still experimental, this approach aims to speed up the natural process of adaptation to changing environmental conditions. Some restoration projects use technology to enhance coral growth physically. Biorock or electromineral accretion systems apply a low electric current to submerged metal structures. The electric field causes minerals such as calcium carbonate and magnesium hydroxide to precipitate onto the frame, creating a favorable surface for coral attachment. Corals grown on biorock structures often exhibit faster growth and higher survival rates, especially in polluted or stressful environments. The electrical current also enhances the deposition of skeletal material, reducing the energy corals need for calcification. Such methods have been deployed in Indonesia, Thailand, and the Caribbean as supplementary tools for localized restoration. When natural structures are lost, artificial reefs can provide new surfaces for colonization. These may be made from concrete, limestone, or biodegradable materials designed to mimic natural reef textures. The goal is to recreate microhabitats where corals, algae, and invertebrates can settle. Modern designs incorporate ecological engineering principles, adjusting surface roughness, chemical composition, and shape to encourage settlement and reduce sedimentation. In some cases, old ships or decommissioned oil platforms are converted into artificial reefs, creating vertical relief that attracts fish and coral larvae. Although artificial reefs cannot replace natural ones, they can serve as stepping stones, aiding larval dispersal and ecosystem recovery. Reef recovery is impossible without addressing water quality. Efforts to reduce nutrient runoff, improve sewage treatment, and manage coastal development have proven crucial. Mangrove and seagrass restoration along coastlines acts as natural filtration systems, trapping sediments and absorbing nutrients before they reach the reef. Community-based waste management programs, particularly in small island nations, help limit pollution and promote stewardship. Without clean water, even the most advanced coral restoration projects will fail. Recent research has shown that manipulating the coral microbiome can improve coral health and resilience. By introducing beneficial bacteria, often called probiotics for corals, scientists can enhance coral immune responses and reduce susceptibility to disease. Field trials have demonstrated that corals treated with specific microbial consortia recover faster after bleaching and exhibit higher survival rates under thermal stress. This approach, still in early stages, represents a promising frontier in biological restoration. Microbiome engineering aligns with the natural function of corals as holobionts, focusing on restoring microbial balance rather than altering genetic structure. As discussed earlier, sound is an important cue for reef life. Restoration projects now use acoustic enrichment, playing recordings of healthy reef soundscapes near degraded areas. These sounds attract fish and larvae, increasing biodiversity and promoting natural recovery. Field experiments in Australia and the Maldives have shown that acoustic playback can double fish abundance and accelerate coral settlement. This non-invasive method demonstrates how understanding natural communication systems can lead to innovative conservation tools. Beyond active restoration, scientists are safeguarding coral diversity through genetic banking. Samples of coral tissue, sperm, eggs, and larvae are cryopreserved in liquid nitrogen for long-term storage. These repositories serve as genetic libraries that can be used to restore populations in the future or reintroduce extinct genotypes. Projects such as the Coral Biobank and CryoReef Initiative are creating global networks of preserved coral material. This approach recognizes that some species may not survive near-term environmental change, but ensures their genetic information remains available for future restoration. Successful reef restoration depends not only on science, but also on people. Local communities living near reefs often rely on them for food and income. Engaging these communities in conservation provides both education and economic incentives. In many regions, citizen divers, fishers, and students participate in coral gardening, monitoring programs, and beach cleanups. These activities foster a sense of ownership and responsibility, reducing destructive practices and encouraging sustainable resource use. Traditional ecological knowledge also plays an important role. Indigenous coastal communities have managed reef ecosystems for centuries through seasonal fishing closures and taboos that allow natural replenishment. Integrating these cultural practices with modern science strengthens conservation outcomes. International cooperation is essential because coral reefs cross national boundaries. Organizations such as the Global Coral Reef Monitoring Network, GCRMN, and the International Coral Reef Initiative, ICRI, coordinate research, data sharing, and policy recommendations. Global agreements like the Paris Climate Accord indirectly support reefs by targeting carbon emissions that drive ocean warming and acidification. Regional efforts, such as the Coral Triangle Initiative in Southeast Asia, focus on protecting the world's most biodiverse reef systems through shared management plans and enforcement strategies. The success of these networks depends on consistent funding, political will, and the integration of local perspectives into global frameworks. While challenges remain immense, the science of coral restoration is advancing rapidly. From microbiome manipulation to acoustic enrichment and genetic banking, a multidisciplinary approach is emerging. The goal is not to recreate reefs exactly as they were, but to help them adapt to a changing world. Resilient reefs of the future may look different, dominated by heat-tolerant species, supported by engineered structures, and managed through adaptive strategies that evolve with new data. Yet their ecological role will remain the same: to shelter marine life, protect coasts, and sustain human livelihoods. Healing the reef is not a single event, but a long-term process that combines science, patience, and cooperation. By restoring balance between nature and human activity, between technology and ecology, we give coral reefs a chance to continue their slow, enduring work, building life from stone, one polyp at a time.